All right, so part one is the lactose system. And so first we're going to just get into lactose metabolism a little bit. So lactose is a sugar, so it's a sugar that you can add to minimal media and bacteria can grow from it. So in order for bacteria to grow using lactose as the primary sugar source, it has to convert lactose into energy, and it also has to be able to turn it into all of the nucleotides, amino acids, fatty acids, etc. Anything that has a carbon source, that lactose has to be turned into. So lactose is what's called a disaccharide. So a disaccharide is a combination of two sugars that are covalently bond together. So this lactose is a combination of galactose and glucose. Galactose and glucose would be called monosaccharides. And so it's easy just to see this as a picture. So one of these hexagonal rings, so we have these sugar rings. So there's 6 carbons, there's 5 carbons and an oxygen, 1, 2, 3, 4, 5 carbons, and then we also have this oxygen that forms this ring. So one of these is glucose. I believe it's this one, but I could be wrong about that. This one would be glucose, and this one is galactose. And these are covalently bonded together at this oxygen molecule right here. So in order for the bacteria cells to metabolize and catabolize lactose as a sugar source, so to be able to survive on lactose alone, so lactose is a sugar found in milk, it has to be able to first cleave this bond so that it separates this disaccharide into glucose and galactose. And then at that point, the enzymes that are responsible for catabolizing glucose and galactose can operate on these sugars in order to form, all of the molecules that are needed. So one of the things that's really cool about this system is that in general, lactose is a very rare sugar for bacteria. They don't encounter it very often. And so they do not want to expend the energy to have the enzymes that are necessary for metabolizing it. So initially, those enzymes are present at a very low level in the cell. And then in the presence of lactose, that induces the expression of those enzymes. So that if you could count the number of proteins within the cell, you would go from dozens to perhaps hundreds to thousands to 10,000s of those proteins. So lactose is an inducer because it results in the upregulation of certain enzymes. And so researchers, 50 years ago actually identified the structural genes that are necessary for metabolizing lactose, and they found that they were all clustered together in a single operon, and this is called the lacOperon. So just as a reminder, bacteria are kind of unusual compared to eukaryotic animals in that they can actually express multiple genes in a single mRNA. And so this lacOperon has a promoter operator region. So this is the regulatory region that controls the transcription. And then it has a transcriptional start site for the mRNA. So the mRNA will come in here, the RNA polymerase will come in here and it'll start to create an mRNA. And so this mRNA will be transcribed and it'll contain the sequence, the coding sequence for the lacZ protein. It'll continue along and it will contain the coding sequence for this lacY protein, and it'll continue along for the lacA protein as well until it reaches the transcriptional terminator, and then the mRNA stops. So the ribosome is actually able to take this single mRNA and it's able to produce proteins for all 3 of these genes. So this is an operon. So it's also called a bicistronic or a polycistronic regulation. So initially there are very low levels of these proteins. However, when lactose is in the environment, if you gave it minimal media plus lactose, or if it's the natural environment in the gut or in plants, it comes across these sugars, then all of a sudden you turn on the expression of these genes. And so you modify the activity of the polymerase, the RNA polymerase at this region right here. So what do these genes do? So lacZ, so the first gene in the operon, encodes a beta-galactosidase. So we've encountered this protein before. And this is an enzyme that converts disaccharide to the monosaccharides glucose and galactose. So this enzyme is able to cleave this bond so that this lactose is now just two monosaccharides, D-galactose and D-glucose. And as a result, this conversion allows for the other enzymes that know how to metabolize galactose and glucose to operate. So, it's a pretty simple enzyme that just simply cleaves a bond. So we've encountered this lacZ gene before, so hopefully you remember this from the cloning chapter, the genetic engineering chapter, and so this is useful for blue-white screening. The second gene encodes a permease, lacY. And so this permease is an enzyme that facilitates the entry of lactose into the bacterial cell. So lactose is a sugar and it's normally in the environment, but it's not able to diffuse across the cell membrane. So each bacteria cell has a lipid bilayer. So this is a hydrophobic region that prevents anything hydrophilic from diffusing across the cell. And so this is a protection for the bacteria to prevent things that they don't want into the cell from entering into the cell, and it also allows them to hold on to the things that they want to keep in the cell. And so in order for lactose to actually go into the cell, it has to be transported through the membrane into the cell. And so this is the job of the lacY permease gene. So it's a transporter that transports lactose into the cell. Once it's into the cell, the lacZ beta-galactosidase can come in and it can cleave this into glucose and galactose. And then the final gene is lacA. So this encodes an enzyme that's a transacetylase, and it's not really well understood. So it's thought to be involved in the removal of toxic byproducts of lactose digestion. So in the process of metabolism, you have the normal reaction, but you also sometimes have reactions that create other chemical species, and these can be toxic. So it seems like lacA is used just to get rid of those. So if you want to use lactose, the lacZ gene and the lacY gene are essential. So these would both be lac minus phenotypes. Whereas lacA, if you knocked out this gene, if you had a mutant in this gene, the cells are still able to utilize lactose as a sugar source. And so the other gene that's within this region is encoded by a gene called lacI. So it happens to be right next to the lac operon, but it is expressed by a completely independent mRNA, and a completely different promoter region is responsible for the expression of lacI. So this is called the repressor gene, and it is what's responsible for having this regulated transcription of the lac operon. So the reason this operon is only expressed when lactose is present is because of this protein right here. So it produces a repressor molecule, it's a transcriptional regulator, and it is able to regulate the transcription of these three genes. It is also known as an allosteric repressor, and so that means it's able to interact reversibly with another molecule, and so this is what provides the regulation of the gene of the lac operon. So if it binds to the second molecule, it causes a conformational change in the shape of this protein that regulates whether or not it can bind to the promoter region. So how does this work? So the lac operon, so we create this mRNA. This mRNA is a polycistronic mRNA because there's three different genes that are encoded, and then the ribosome can come in and translate all three of these genes into unique proteins. So there's three unique proteins that would be created from this operon. So the lac operon is regulated by the presence of lactose. So if you don't have any lactose, the lac operon, there's no mRNAs, or there's very few mRNAs. And then if you look at the lac operon in the presence of lactose, you see a bunch of it. lacI, the repressor, this is expressed by an independent promoter, and this promoter is not regulated by lactose. So lacI, this repressor protein, this is around no matter what. So it's around if there's lactose around, it's around even if there's not lactose. So just remember there's two different promoters, there's two different mRNA, and they're regulated in different ways. So in 1960, Jacob and Monod proposed a Operon model that explained how this negative control can occur. And so what they proposed is that the lacI gene regulates transcription of structural genes by producing an allosteric repressor molecule, and this Operon is subject to negative control. So transcription occurs only when the repressor fails to bind. And the repressor normally binds to DNA sequence, and it inhibits the RNA polymerase and represses transcription of the structural genes. So let's see how this is thought to occur. So this repressor gene, this is around all of the time. So because this promoter is always expressed, you always have this repressor molecule around. And so this repressor can bind to a small molecule. And so in this figure, it's called lactose, but in point of fact, it's actually a molecule called allolactose, which is a derivative of lactose. And so if lactose is present, then allolactose can bind to the repressor protein at a specific site. So there's two different states this lacI protein can take. It can either be bound to allolactose, or it can be not bound to allolactose. And so if it's not bound to allolactose, then there's a shape change that allows this to bind to DNA. And if allolactose is, then it's no longer able to bind to DNA because of the shape change of the protein. So this is what's referred to as the allostery. So in situations where no lactose is present, this repressor molecule is in the form where it can bind to DNA. And so where it binds is a region upstream of the promoter called the operator region. So there's a DNA binding site for this. And the action of this repressor binding to this region prevents the polymerase from being able to initiate transcription. So you have no transcription in this case, and so that's why the Operon is turned off. But now we add lactose to the environment, and so now this repressor molecule has this modified shape because it's bound to allolactose. And so now it's unable to bind to DNA, and so this repressor molecule remains in the cytoplasm. And so now the polymerase is able to bind to the promoter region and create the mRNA that's necessary for the translation of these proteins. So in this way, you're able to convert the presence or absence of a sugar to transcriptional control because you have this special protein that's able to bind to the sugar. And so just to go over what I said, so lactose, this is the sugar, which is a combination of galactose and glucose. And this beta-galactosidase typically cleaves this bond right here, but it can also actually do this alternative reaction, which results in a change of what the covalent bond is. So this covalent bond between the oxygen now forms with one of the carbons in the sugar. And so this is what's referred to as allolactose. So if lactose is in the environment, it's transported into the cell by the permease. The beta-galactosidase acts upon it to cleave most of it, but some small amount is converted to allolactose. And then this allolactose is able to bind to the lac repressor, and then that prevents it from binding to the DNA. And so the lac Operon is able to be transcribed. There's some other chemicals that are known to actually be able to bind to the lacI repressor protein, so IPTG. This is kind of the molecule that Monod used, Jacob and Monod used for many of their experiments. It's called IPTG. And then for the blue-white screening, there's another derivative called XGAL. And so this XGAL is what gives the bacteria color. And so the beta-galactosidase can cleave, and then the result of this beta-galactosidase cleaving is the result of this cleaving is whether or not there's blue or white bacteria. So this is how we can use beta-galactosidase as a indicator of whether or not the cloning reaction occurred successfully. So this is the model that they proposed. So it invoked a series of molecular interactions between proteins, inducers, and DNA. So the allolactose binds to the protein, and the protein binds to the DNA, and that's how this regulation can occur. So in the case of no lactose, if lactose is not present, the enzymes are not needed, and expression of genes encoding enzymes is repressed. When lactose is present, then we need the lacZ, the lacY, and the lacA, and so it indirectly induces activations of the genes by binding the repressor, and this is negative control. Once all of the lactose is metabolized in the environment, then none is available to bind the repressor, and then transcription is repressed. So this was just a model, and so in order to test a model, you need to have certain predictions. And so one of the predictions is that the I gene produces a diffusible product, and so this is referred to as a trans-acting product. The O region, the operator region, is involved in regulation, and so this region must be adjacent to the structural genes to regulate transcription, and so this is referred to as a cis-acting element. And so how do they test this model? There's two different primary ways. So one, they isolated mutations in different parts of the lac locus, and they assayed the effect on the regulation. And then they also created partially diploid bacteria in order to compare the cis versus trans regulation. And so we'll see how they did that in a second. So just remember, a marrow-diploid bacteria, typically the way you create that is that you have a plasmid that also contains a gene or a part of DNA of interest. All right, so let's see how they tested this. So one way they tested this is that they identified a mutation in the lacI gene. So the lacI gene is no longer functional, and so the lacI repressor is no longer able to bind to the operator and induce transcription. So what is the prediction? The prediction is that we have constitutive expression. So no matter whether or not you have lactose around or lactose not around, we expect to always see transcription of the lac operon. You can also have mutations in the operator. So the operator site, that's just DNA. And so you can also have mutations here, and if these mutations prevent the repressor from binding to it, so this repressor recognizes certain sequences of DNA, and so if those sequences are mutated, then this repressor is no longer able to bind to it. And so the prediction for that, again, is constitutive expression, because the polymerase is always able to act and create the mRNA. You can also induce gain-of-function mutations. So gain-of-function mutations are the opposite of loss-of-function mutations. So the inducer, in this case, instead of being unable to bind to DNA, now it no longer needs the lactose in order to adopt the conformation that allows it to bind DNA. So before we talked about the effect, the idea that you had two conformations of the lac repressor. So one is when the allolactose is around. So if allolactose binds to it, then the conformation changed, so it's not able to bind to DNA. And so the idea behind the constitutive mutation is that you now have a version of the protein that has lost that allosteric regulation. So even if allolactose is bound to it, it's still able to bind to that operator site. And so in that case, repressor is always bound to the operator, blocking the transcription. And so then the prediction is repression. So you never see transcription of the lactose system. And so, these were the predictions, and these predictions were validated by various experiments. So these are the various genotypes that they tested. And they also, this is the wild type expression. So if lactose is around, you see transcription. If it's absent, you don't, so it's turned on. They also tested, for example, what happens if you don't have the lacZ gene. And so this will be part of the activity to think about this, to think this through. But many of their predictions were actually true. And so the final thing they tested were what are called marrow-diploid strains. So again, this is when you use the F factor, the F prime factor that actually contains parts of the DNA from the chromosome. And so they also tested the ability of this marrow-diploid to rescue it. Can providing the lacI gene on a plasmid rescue the effect? And so again, it's a diploid because you have two, it's a partial diploid because one gene has two copies. So we have two copies of the I gene. So you have a wild type copy and you have a minus copy. And so the wild type copy, you have this plasmid and all it contains is the lacI gene. So it does not contain, so an important note is that it does not contain any of the other Operon. So all this plasmid contains is the lacI gene. But we can see that this is able to rescue. And what do we mean by rescue? So we start with this I minus O plus Z. So, we start with this right here, which is plus plus. And now when we provide this plasmid, we have plus minus, which is the wild type phenotype. What if we do the same thing with the operator? So we started with the constitutive operator site. And so this right here, you always have expression. And so what if we now create a plasmid that contains the wild type operator site? Okay, so the DNA of the operator site, but you do not have, so you do not have the lac Operon. So like this lac Operon that you normally have on the chromosome, this is not on the plasmid. All you have is this operator region. So it'd just be having this little piece of DNA on there. And so this is not able to rescue. And this is what we mean by the difference between a trans-acting factor and a cis-acting factor. So the trans-acting factor, it creates this plasmid, creates a protein product, which can then rescue. Where in this case, this operator does not produce anything that can diffuse through the cell. So the only way for this operator to have an effect is that it has to be next to the lac Operon. That's the only way this thing can have an effect. So this is what is a cis-acting factor. And so again, as part of the activity, you also will go through kind of these other different types of genotypes and try to predict what's going on. So hopefully, what we've gone through will allow you to this is a conceptual chapter. You have to understand the concepts of how the system works. But then once you understand the concepts, hopefully you'll be able to figure out, what a genotype will do just by kind of, following the logic of it. So that is the end of chapter part 1, and we will go into the CAP system